We often picture our DNA as a highly secured, architectural blueprint locked safely away in a vault. But the environment inside a cell's nucleus is far less peaceful. It is an active war zone, and your genetic code is constantly taking hits. This diagram shows a baseline estimate of the daily destruction. In a single standard human cell, the DNA structure experiences at least 10,000 damaging events every single day. If these microscopic injuries are ignored, structural errors begin to accumulate. This steady accumulation is the direct root cause of cellular aging, senescence, and ultimately cancer. Survival against this massive daily onslaught depends on a hidden, highly sophisticated hierarchy of cellular repair crews, working around the clock to fix damage before it becomes permanent. The attacks come from two distinct fronts. First, there are unavoidable internal threats, like the reactive oxygen species produced as a simple byproduct of keeping your cells alive. But then there are the severe external threats. Looking at this archival photograph of an industrial factory, you can see the heavy plumes of exhaust. We are consistently exposed to mutagenic chemical compounds through this kind of industrial pollution. We also introduce these severe mutagens voluntarily. The thick cloud of tobacco smoke you see here acts as a highly efficient delivery vehicle, driving severe chemical damage straight into our tissues. And even a routine trip outside carries immense risk. Every person sitting on this crowded beach is actively subjecting their skin cells to a relentless barrage of ultraviolet radiation from the sun. Defending the blueprint of life against this relentless combination of internal metabolism and harsh environmental exposure requires a specialized biological toolset. Different scales of damage require distinctly different repair mechanisms. The first line of defense handles the smallest scale of errors, simple typos. When DNA replicates, the polymerase enzyme occasionally matches the wrong bases, for example, sticking an adenine across from a cytosine. To fix this, the cell deploys mismatch repair, or MMR. A single base pairing error distorts the helix structure. MMR proofreader proteins constantly patrol newly built DNA, specifically feeling for this protruding bulge. Once located, they cut out the erroneous stretch. A polymerase drops in the correct base to restore the geometry, and a ligase enzyme seals the gap. By catching these minor misprints immediately after they happen, this meticulous proofreading system prevents routine biological typos from locking in as permanent genetic corruption. The scale of damage escalates when dealing with chemical alterations caused by our own internal metabolism. For small, non-distorting chemical changes to single bases, the cell uses base excision repair, or BER. This pathway relies on specialized enzymes called DNA glycosylases. Think of them as targeted spot clairs that continuously scan the vast genomic sequence, searching for very specific, chemically altered bases. The extraction process is remarkably physical. When the glycosylase enzyme identifies the damaged target, it grabs the individual base, flips it entirely outside the helix structure, and severs it from the chain. With the damaged unit removed, a separate group of enzymes clears away the now empty sugar phosphate backbone, making room for a fresh, pristine nucleotide to be inserted. Base excision repair operates as a high-fidelity maintenance routine. It systematically neutralizes the toxic byproducts of our own metabolism before they can accumulate and trigger cellular disease. Now, let's step up the scale of destruction. When you stay outside too long and get a sunburn, what is actually happening to your DNA on a mechanical level? UV radiation causes bulky, helix-distorting damage. These are pyrimidine dimers, two adjacent bases abnormally fusing together. To fix this, the heavy-duty nucleotide excision repair system steps in. Proteins unwind the area, cut the strand on both sides, and remove a massive 32 nucleotide section, leaving a cavernous gap. Once the bulky damage is completely cleared out, DNA polymerase uses the untouched opposite strand as a clean structural template. It synthesizes a brand new line of DNA, perfectly patching the hole. Without nucleotide excision repair serving as an essential biological barrier, everyday sunlight exposure would rapidly overwhelm our skin cells and guarantee skin cancer. All the mechanisms we've covered so far rely on one crucial factor: keeping at least one side of the DNA strand intact to use as a template. but high-energy ionizing radiation can sever both strands at once. We can easily generate this level of destructive energy. Looking at the heavy mechanical components of this vintage X-ray equipment reminds us of the sheer power of exogenous ionizing radiation. A double-strand break is an existential crisis for the cell. Because both sides of the helix are severed, the perfect repair template is entirely gone. The chromosome is literally falling to pieces, with the severed ends drifting entirely apart. This biological emergency forces the cell into a desperate calculus. It must immediately weigh the need for exact genetic precision against the strict urgency of pure survival. The ideal solution is homologous recombination, or HR. It is an entirely error-free fix. As this diagram illustrates, if the cell happens to have an identical, undamaged sister chromatid nearby, it can use that sister strand as a perfect structural template, flawlessly copying over the missing sequence blocks. But a sister chromatid is only available during very specific phases of the cell cycle. If a double-strand break happens without a template available, the cell has to deploy its emergency responder, non-homologous end joining. This process is incredibly fast and messy. The cell rapidly grabs the broken ends and glues them back together completely blind. Without a template to guide the repair, base pairs are randomly added or permanently lost at the jagged seam. The cell accepts this chaotic, mutation-causing patch job for one simple reason. A mutated chromosome is better than a shattered one that leads to immediate cellular death. Whether it's meticulously proofreading a single typo or desperately gluing together a severed strand, our survival is defined by this endless, active process of biological repair.